The overall goal of this procedure is to quantitatively evaluate the respiratory muscle activation patterns in individuals with spinal cord injury through comparison with normative values obtained from healthy subjects. This is achieved by recording of electromyographic, or EMG, activity of respiratory muscles in individuals with spinal cord injury during respiratory tasks to calculate the amount of such activity for each muscle. As a second step, a resultant vector is constructed which characterizes the distribution and amount of activity across these muscles. Next, data from healthy subjects during the same task is used to calculate a prototype response vector. A similarity index can then be calculated that relates the degree to which the resultant vector resembles the prototype response vector, allowing a comparison between the respiratory muscle activation pattern in patients with spinal cord injury to the normal pattern that would be observed in a healthy subject.
Using this technique, we can monitor respiratory motor function in patients with spinal cord injury or other disorders associated with respiratory motor control deficits. Data acquisition and processing is very important for successful evaluation of this method. That's why the visual demonstration is important. Demonstrating the procedure will be Sevda Aslan, research scientist, Edward Brown, research manager, and Manpreet Chopra, research technologist from my lab.